Good evening and welcome everyone. On behalf of the Del Mar Foundation, I am delighted to welcome you to this evening's DMF talk and to introduce our two speakers. Both of them are from Sanford Burnham Prebys Medical Discovery Institute, and they join us this evening to bring their up-to-the-minute perspectives on a topic of great concern to all of us, COVID-19, a light at the end of the tunnel. Our invitation for this talk included a short bio of each of our distinguished speakers, so I'll offer just a few words of additional introduction. Dr. Evan Snyder is Director of the Center for Stem Cell and Regenerative Medicine at Stanford Children's Health Research Center. He has been a leading researcher in stem cells and regenerative medicine for over 20 years. The Snyder Lab's perspective, as stated on its webpage, strikes me as wise and applicable to this evening's topic as well. So I'll introduce Dr. Snyder with his lab's stated perspective. Stem cells offer an intriguing mix of controversy, discovery, and hope. Politicians are charged with dealing with the controversial facts of stem cells. We prefer to focus our energy on their potential for discovery and hope. With respect to Dr. Sumit Chanda, Director of the Immunity and Pathogenesis Program at Sanford Burnham Prebys, I'll introduce him in the spirit of, what have you done for us lately? Today's Twitter post from Sanford Burnham Prebys, posted at 8:59 this morning, announced the following that happens to relate directly to this evening's topic. The tweet reads as follows. The Chanda lab has found the sensor that detects SARS-CoV-2 and signals it's time to mount an antiviral response. It's an RNA binding protein that acts like an immune cop and calls for backup. The research opens new avenues to treat and prevent COVID-19. You can read this morning's full press release at bit.ly/immunecop. If you have questions for our speakers, please post them in chat. You'll find a chat button somewhere near the bottom of your screen usually, and we'll ask as many of the questions as possible at the end of the speakers' presentations. Please make sure that you are muted once the presentations begin. And if you wish, you may also want to stop your video and of course, use the speaker view to get the best possible view of the presentation. And now I know that you all join me in giving a warm Del Mar welcome to Dr. Sumit Chanda and Dr. Evan Snyder.
Hi all.
Hi. So Betty, did you want me or Evan to go ahead and start on the
Your
presentation?
choice.
Why don't you
Okay,
start?
I'll go ahead and start. So thank you for that introduction, Betty. And I actually had no idea that Twitter feed came out. So it's good to know that our work is getting out there. So I put together a few slides to share with you about what essentially the last year or so has been like for me and the folks in the lab. And I'm not going to talk about the immune cop story, but another story that we've been working on dealing with trying to discover SARS-CoV-2 antivirals through a strategy called repositioning or repurposing. This means taking old drugs and seeing if they can have new purposes. So just to give a background on how we got to where we are now in the world, it likely all started from a single bat somewhere outside the Wuhan or within the Wuhan province of China. It is thought that, let me see how we forward it. So the bat transmitted the SARS coronavirus to humans, either directly or through an intermediate species called the pangolin. So there's some genetic evidence that there was an intermediate species that led to the transmission into humans. And what we don't know right now is if SARS-CoV-2 had software preloaded to be able to be transmissible from human to human. or that's something that developed, and we're seeing that develop even further now with the emergence of the UK strain and the South African strain. Beyond humans, we know that we can pass the virus on to various different animals. We just heard that there were 2 gorillas in the San Diego Zoo that were infected with COVID. So this is an important thing to keep in mind because even if everyone gets vaccinated, there are going to be reservoirs of the virus lingering around. And so you'll see that my thesis, my hypothesis is that we're never going to eradicate SARS-CoV-2, but it should become endemic like the flu, like other coronaviruses. We can talk a little bit about that. So this is a little bit of an old slide, but these are the viruses out there, respiratory viruses that cause pandemics. So flu is on the left side, and then the coronavirus is COVID-19, and then prior to that we had SARS and MERS. There's a couple of different important points about COVID-19 that really allowed it to hit the sweet spot from a virological perspective of becoming a pandemic virus. One is this R0 coefficient. So R0 just a fancy way of saying for every person that gets infected, how many people does that virus spread to? So flu is relatively low. The original SARS was higher. It's unclear what the new variants R0 is going to be, but they're saying it's somewhere between 50 and 70% higher than the old variant. So the R0 is very important, but the other thing is the incubation time, right? So the thing that makes SARS-CoV-2 or COVID-19 particularly insidious is that you have this period of asymptomatic infection where you're spreading the virus without knowing you're sick. If you know you're sick, you can stay home, and so that limits the spread of the virus. Things like viruses like the original SARS or flu have a much more compressed window. So this expanded window really allows SARS to be spread before it becomes detected and people can stay home and quarantine. And then there's the case fatality rate. Right now, with the vaccines and drugs, case fatality rate for flu is between 0.05 and 1%. So this still represents somewhere around 30 to 50,000 deaths a year. This is still a major health problem, but it doesn't stop us from going to restaurants, going on in airplanes. And so the goal for COVID-19 is really going to be to get that R0 down through vaccination and get case fatality rates down through both vaccination and therapeutics. If we want to look back at the last great global pandemic, we can look at 1918. And on the left-hand side here is the impact of the 1918 influenza pandemic on life expectancy in the US, where you can see that World War I and World War II had a negligible impact on life expectancy, whereas the 1918 influenza pandemic dropped it by about 10 to 15 years. On the right-hand side, you'll see that much like what we're experiencing now, the 1918 pandemic was not a single event. It came in 3 different waves. One was an early spring wave that was fairly diminutive. Next was the more rampaging winter wave, which is what we're in right now. And then a third wave that happened in late spring of 1919. There's no reason to believe that SARS-CoV-2 is not going to follow the same pattern, but hopefully with the rollout of some of these vaccines, we can flatten that last wave curve. We obviously haven't been able to do that with the second wave. But I'm optimistic that by the time we get to the third wave, it'll look more like things did back in last April and May. So the other thing to remember from the 1918 pandemic, which killed 500,000 people here in the US, is that it never went away. It became seasonal. And this had nothing to do with vaccines. Vaccines came out in the 1930s, right? And so by 1919, 1920, the virus had gone from being these various waves of infection to just coming back every season and exacting tolls like normal influenza does. But the other thing to learn from this epidemic or this pandemic was that there are predictable, not predictable, but frequent pandemics of influenza viruses that are caused by reassortment, or in comparison to coronavirus, those would be caused by mutation. So flu changes itself enough where it can then start a new pandemic again. So in 57, we had another pandemic. In 68, we had another one. In 77, we had an emergence of a new virus, but because it was so much like the 1918 virus and people had pre-existing immunity, it did not cause a pandemic. But most people can remember back in 2009, I think we really dodged a bullet with the swine flu here. It did represent a pandemic virus, but it wasn't as lethal as we once feared. But we would expect that this kind of trend will happen now with SARS. Now we're on SARS-2. There's a reasonable likelihood that different coronaviruses will continue to emerge every 20 or 30 years. And it's very important that, along with flu, we really become prepared for it and really take the lessons of 2019 and 2020 to heart, and not anticipate that we're set for the next 100 years. So how did we end up getting into our research? So this started a little over a year ago. Xin Yin, who's an incredibly talented postdoc in the lab, went home to China for a very well-deserved Christmas break. And he started emailing me about a pneumonia with unknown etiology that was breaking out in the Wuhan province in China. And then he emailed me that they had isolated a virus that appeared to be the causative agent for that virus. So I told him to come back immediately, and we are virologists, and we kicked into high gear. Anytime you hear the words novel and coronavirus, or novel and influenza virus together, as a virologist, warning bells go off. And so we put together a dream team of virologists, including Ren Sun, Kwek Yun at Hong Kong University, who discovered the first coronavirus, and Adolfo Garcia Sastre at Mount Sinai, and teamed up with Scripps, who had, through funding from Bill and Melinda Gates Foundation, put together a library of approximately 12,000 known drugs. So these are drugs that have either been approved by the FDA or have been in clinical trials. And so typically it takes anywhere between 10 to 15 years to discover a new drug. But it's much quicker to take an old drug that we know has good safety data and see if it can work on a new disease. And this is what's called repurposing or repositioning. And our initial idea was to take this library of 12,000 drugs, along with Lara Riva, who was a researcher in the lab. She was going to fly to Hong Kong because they had the virus, and we couldn't get the virus out of China at the time, and help them screen through these libraries to find potential treatments for coronavirus, this novel coronavirus. But in early February, there was a travel ban instituted in place, and so Laura couldn't get out there. So we had to quickly pivot to a plan B, which was doing drug discovery by iPhone. So we used an app called WeChat, and then we shipped a bunch of automation, shipped the library, shipped a bunch of things from the lab out to Hong Kong. Of course, shipping during a pandemic well, shipping in general is never easy, but during a pandemic, definitely harder. Our packages didn't end up where we wanted them to. These were all things on dry ice, so we started panicking. But fortunately, when the boxes arrived, they had refilled the dry ice. And there you see some of our instrumentation behind what's called a BSL-3 cabinet, or biosafety cabinet, which you have to use SARS-CoV-2 in. So you use this app called WeChat. Lara would get up in the middle of the night to coincide with Hong Kong time, and essentially talk them through how to do this drug discovery campaign by using her iPhone. And so Kim Tingley of the New York Times Magazine did a great write-up on these guys who did this work are heroes. So if you want to really get an idea of the mountains that they moved in order to do this, there's a nice write-up in the May 13th issue of the New York Times Magazine called "Can Team Science Yield a COVID-19 Treatment?" So meanwhile, back here in San Diego, Laura Martin Sancho, another researcher in the lab, was one of the first people in San Diego to receive an isolate of the SARS-CoV-2 virus. And this was the first patient in Seattle, Washington that was diagnosed with the virus after travel to China. And the CDC made this available to a select few researchers in the area. And so what she did was heroically within a week figured out how to culture this virus. Now, typically it takes months to figure out how to culture this virus and figure out a way to assess the impact of different antiviral drugs. So this bottom panel here you see is that each of these little squares have cells in them, and the green cells represent coronavirus infected cells. And as you increase your concentration of drug, you can see that there are fewer and fewer green cells, meaning there's fewer and fewer coronavirus infected cells. So this was a way for us to sift through these 12,000 compounds, these known drugs, to find ones that could work against coronavirus. And so through this, these kinds of heroics, we narrowed things down to about 20 compounds. I won't get into the weeds too much here, but essentially what we're showing here is the activity of these antiviral compounds against SARS-CoV-2 at different concentrations. The yellower they are, the more potent they are. And for you aficionados in the audience, these are what are called standard dose response curves. So this was a first pass assay that we used. And then what we did was develop what we call a down selection funnel. So each step of our process, we got more and more closer to the real infection in people. And I can very quickly talk you through it. But we took those 20 drugs and we had found remdesivir, which is now approved for use in coronavirus treatment in this assay. But it was a nice confirmation that our approach was working. Also found about 20 other things, tested them in things like primary stem cells, iPS-derived stem cells. So these are the lung cells that SARS-CoV-2 replicates in. So Evan will tell you more about iPS-derived cells, but we can show that these drugs are knocking down virus. DMSO is what's considered a negative control. And we're seeing really good knockdown with some of these drugs comparable to remdesivir. Now, you have to remember, these are all drugs that are that we have safety data for, so we can move fairly quickly with these. The next assay we use is we use lung explants. So these are biopsies from patients infected with SARS-CoV-2, and we were able to test some of these molecules and show that the virus can be blocked by these known drugs in these models. So the next step was to then move into animal models. And so I want to highlight one of the molecules that we're very excited about called clofazimine, or the trade name is lamprey. We're excited about it because it's been around for about 40 years. It's been used to treat leprosy. It's an FDA-approved drug on the WHO's list of essential medicines. Why we're excited about it is that two things. One, you could take it as a pill. And secondly, it's cheap. It's about $1.50 a day to take. In contrast to remdesivir, which is about $2,500 a treatment, and there's not that much of it to go around. This is cheap. Remdesivir, you have to go to the hospital to take it. Here, you can potentially give this as an outpatient. Say you are diagnosed positive or have symptoms, just go to your CVS, pick this up, and you can get treatment early. And we know the earlier you treat viral infection, the better your outcomes are. So we did hamster studies. So we did 2 series of hamster studies with this drug. Hamsters seem to be a pretty good model to match human disease. And again, I don't want to get into the weeds, but what we see is when we give this clofazimine compared to untreated, you see a knockdown of, this is viral titers in the lung. And this is when you give clofazimine prophylactically, means before virus infection. And this is therapeutically, which means during viral infection. Remdesivir is in blue. It doesn't do as well as remdesivir, but still does move the needle pretty sufficiently. And it has the added benefit that you can give it much earlier because you don't have to be hospitalized to get this. I won't go through this. This is just the pathology in the lungs and some other markers. The other thing that was exciting was that we found that clofazimine and remdesivir can be used together to synergize. And that means that you can add lower and lower doses of both drugs and you can extend their impact. So essentially what this graph shows, the redder you see, the more synergy you see between the 2 drugs. And I think what really hits home here is in blue here, where my pointer is remdesivir alone. This is a low dose remdesivir. This is a low dose clofazimine. And you can see when you combine them, you see a massive reduction in viral titers in the lung. And I think even more important is looking at nasal washes. So this is how you this is a surrogate marker for transmission. And so remdesivir doesn't do a good job in blocking the amount of virus that you have in your nose. Now, this doesn't affect you negatively from a disease severity standpoint, but obviously that's how things get transmitted. And what we find is that the combination of clofazimine and remdesivir knocks down titers, and this is a log scale, so we're talking by about 10-fold in the nose. So we think that this combination not only can help you from getting severe disease, but can also help stop you from transmitting the virus onto somebody else. So we're very excited about this. We're now applying for funding to initiate phase 2 clinical trials. And because this is a known drug, we can move fairly quickly into these clinical trials and then emergency use authorization, having another antiviral for SARS-CoV-2 that's safe, that's effective, and can work with existing drugs. So that's in a nutshell one of the projects that we've been doing in the last year, and just looking ahead, happy to take some questions going forward. But one of the things I want to point out is that while the current series of vaccines are extremely remarkable, they are the first generation of vaccines. We don't know what their durability is going to be. We don't know essentially if we are going to have to take boosters every year. You have to think of these vaccines like iPhones, right? The iPhone 1 was great when it came out, but the iPhone 11 or 12, I don't know where we are now, is so much better. And these vaccines are going to keep getting better, and we really need to keep working on those, and as well as developing new antivirals. So as I mentioned, my real gut feeling is that we are not going to be eradicating SARS-CoV-2. Smallpox is the only virus that a human virus that we've only successfully eradicated, and that's because there's no animal reservoir. SARS-CoV-2, I think, will be with us for the foreseeable future, but we have to continue the science and the development, both for antivirals and for improved vaccines. And finally, this time we have to now anticipate the next pandemic. We had a series of near misses, and I think a lot of people thought, well, this was the boy who cried wolf. But I think we should now look at those as nature giving us warning shots that we did not heed. And if we do not heed the lessons of SARS-CoV-2, there could be a deadlier, more impactful pandemic. And if we don't continue to invest in the development of new science, new medicines, new vaccine technologies to combat the potential pandemics of the future, I think that we would be missing major lessons that could have been learned during this pandemic. So before I sign off, I just want to thank all of the heroes that in the lab that did the work, notably Laura Martin Sancho, Laura Riva, Xin Ying, our collaborators in Hong Kong, SF Young and KY Yun, Arnab Chatterjee at Scripps, Adolfo Garcia Sastra, Andrew Su at Scripps, and Ren Sun at UCLA and University of Hong Kong. And the funding sources, the DoD, NIH, and NCI. So that's my short presentation. And I think, Evan, you're up next, and then we can tag team on some questions.
That was great, Sumit. Thanks so much. Hopefully you can both hear me and see my slides. Can somebody just give me a thumbs up to make sure that you can both hear me and see my slides?
Yes.
Okay.
Yes.
All right. So that was an excellent introduction that Sumit gave to both the virus and the pandemic. And you might wonder, what's a dumb stem cell biologist doing in a field in which he is completely out of his depth? I'm not a virologist. I'm not an immunologist. Well, I have my own Chinese story. And I happened to be in China the second week of December and became friendly with, obviously, with my hosts, but also started getting wind of something going on in a city that I had not visited, even though I had been visiting and giving talks at a number of cities. But I kept track of that. And when I came back, I stayed in touch with my colleagues in China, and it was becoming clear, one, that this was heavily a respiratory virus, and two, there was just not a lot known about it. And when I came back, more was not known than was known. Now, very fortunately, I don't think it was mentioned, but in addition to being a stem cell biologist, I'm also a neonatologist, a pediatrician, a clinician. Pediatricians, and particularly neonatologists, deal with lungs. And I'm a great supporter of physician scientists, training them, nurturing them. And in the group at the time was my fellow neonatologist on the faculty of UCSD, Sandra Leibel. And she had been growing mini lungs in addition, I'll explain what that means in a second, to try to understand some of the diseases that newborns get, genetic diseases. And then we are expanding into trying to understand lung cancer. And I came back, Sandra and I put our heads together and we said, you know, we have the literature's only true mini lungs in a dish. There are so many questions unknown about this disease, but we're in a position to answer them. Now, coincidentally, just as we were formulating this approach, we had learned that Sumit, completely independently initially, was screening drugs and was doing as virologists do when they want to do high throughput screening of drugs using cell lines that are not actually clinically relevant, but really allow you to get powerful signals, you know, monkey kidney cells and lung cancer cells and things like that. And we all put our heads together and realized Sumit heroically, as you heard, whittled down 12,000 FDA-approved drugs to a handful that we wanted to fast track to the clinic. We had human mini lung cells in a dish that might be able to give it some clinical relevance, that might help fast track these drugs into patients. So this is now around April or March. Let me tell you a little bit about the mini lung so you can understand why stem cell biology will play a role, does play a role in this disease. So obviously, this is a schematic of the lung. There's the top, there's the part that's closer to the windpipe, the trachea, and then there's the distal bits. Each one has its own particular cell type. How do you mimic that in a dish? Well, the way to do that is, or at least the way we decided to do that, was something that is a trick in stem cell biology. You can take cells from a patient, blood cells, skin cells, and then treat them with various chemicals to push them back. Now, before I tell you about that, I want to mention who can you get these cells from whom you're going to make the mini lungs. Well, you can get them from whoever the patient happens to be. And this allowed us to make mini lungs from not only Caucasian populations, but African American populations, Latino populations, Asian populations, men, women, young and old, and various genetic predispositions. Now, why is this important? I'll get to this later on, but I think anybody who's followed the news knows that this disease does not strike in a uniform manner. For all kinds of reasons, there is great disease disparity. Now, some of this may be due to adverse socioeconomic conditions and comorbidities, and that it certainly plays a big role. Some of this could be due to certain genetic predispositions or particular molecules that fight infection in some groups better than others. But we were in a position to try to study that. So now we take the cells from this whole array of patients and we treat them with molecules that then take these cells and push them back to the way they would look if they were in the earliest stages of development in an embryo. And then you simply follow nature's recipe. How did nature make lungs? We start understanding that through development, we do the same thing in a dish. And we make the proximal, the middle, the distal, and we have patient-specific lung organoids or mini lungs in a dish. Now, what we can do in addition to that is try to mimic the various kinds of conditions that comorbidities and other diseases could expose these lung cells to in various kinds of patients, certain toxins, certain differences in the microbiome, certain abnormal kinds of environments like in diabetes. And then in a dish, we follow the development from the earliest stage, but in a dish, the way it would look as the lung develops in the actual patient. So now we have a whole array of lungs. This is an example of what these mini lungs look like. They expand when you give them the right developmental molecules. Now, I won't get into this, but it's very interesting that this last stage where the cells balloon, where the little mini lungs balloon up, is in response to a molecule, to a drug called dexamethasone. That's a steroid that's known heavily in development. It's also anti-inflammatory, but anybody who's followed the news knows this is the drug that seems to be one of the most powerful drugs in treating patients. The mini lungs also could have little bits of smooth muscle. So maybe you can see in this movie, little respiratory movements, almost as if they're contracting. And then under some circumstances in some cells, you can even see these beating little cilia on cells that would emulate another kind of cell that normally in the body tries to clear debris or clear infection. Now, I think you've learned, and the field has learned, that COVID-19 is not solely a disease of the lung cells per se. It's also a disease of the blood vessels that feed the lungs. It's also an inflammatory disease, and there are inflammatory cells called macrophages that make these kinds of inflammatory cytokines that in and of themselves, when appropriate, can cause damage. And COVID-19 is a whole array of all of these kinds of diseases. Well, in these mini lungs, we can take these stem cells, these induced pluripotent stem cells that I show you about, and not just make them lung cells, we can make them blood vessels, as you can see over here. We can make them macrophages and invest them so that we really do have not just a mini lung that just has lung cells, but all these other aspects to it. Now, thanks to this collaboration with Sumit, Sumit's group created SARS-CoV-2, a pseudovirus. Now, a pseudovirus is the virus that binds to the cell but doesn't cause the damage. It allows you to look at the very first stages of the infection. Now, this is good because when you want to study what cells are going to be infected, how do they get in, what's the receptor they use, you don't want to throw a hand grenade into the whole culture, into the whole mini lung. You want to break it down into its little steps, and this allowed us to do it. So the pseudovirus is green, and you can see that this little mini lung up here in this upper right-hand corner got infected. And we can then slice and see what are the cells that are getting infected. Now, it's interesting that not until we got complete differentiation, really firmly being lung cells, did we get infection, as you can see by this spike over there. Well, that's great. These are cells that are heavily green. They've heavily been infected. Now we're set up to sort of see what drug is going to block that. And now that's what we do. First, we can take, as thanks to Sumit, who has winnowed down a list to a few drugs, we can take some old FDA-approved drugs and find out which ones really do block this infection. And as you can see in this column over here, that this repurposed old drug blocked the infection. Up here, heavily green, down here, dramatically diminished green, dramatically diminished infection. But then, of course, thanks to Sandra, we can use complete, active, living virus, as you can see over here, and then come up with even new drugs as we get to know better and better how this virus lives. And here we have 2 new drugs that also seem to block infection in these lung organoids. Now, I'm going to take up a point that Sumit made, and this can't be emphasized enough. A complex disease like this needs a whole armamentarium. And thankfully, we have these vaccines, and they're going to get better, as Sumit mentioned. But even after we have available vaccines, we will still need drugs to be part of our anti-COVID-19 armamentarium. First of all, we still need to figure out, is the immunity sufficiently broad and complete and lasting for all patients? There are other pathologic processes other than just infection, and I mentioned some of them that, oops, doesn't look like it wants to go. For example, inflammation. And this is heavily a blood vessel disease, a vasculitis, a vasculopathy. That's why you see stroke, and that's why you see all kinds of other inflammatory things going on with kids. And then this virus can enter the cells by other routes, not just this spike protein that goes onto this one receptor. It's a very clever virus and can find alternative ways of getting in. And we need to figure out how to block those entry paths, even if we block the primary preferred entry path. And then, of course, what we need to do is make sure when we come up with a good vaccine or a good drug or any other treatment, that it is amenable, it is useful for all, regardless of your race, gender, ethnic background, age, comorbidities, genetic predisposition. Maybe it simply means that we play with the dose. Maybe it means that we put together synergistic drugs. Sumit talked about remdesivir and cofloxamine. maybe we need to figure out how to do that. Well, we're in a position now to try to address disease disparities so that the treatments that work for everybody. Now, I'm going to end with this because I was asked to just comment on this, and I won't spend a long time here, but maybe the Q&A, we will get into this. This is what I did during my Christmas vacation. I think I had mentioned that I'm a clinician. I think you're aware how bad this disease is hitting this state. This is a true public health emergency. And it is really true that the hospitals are being completely overrun. So at the beginning of the pandemic, I actually volunteered to join California's Emergency Medical Services Authority, or it's also called the California Medical Assistance Team. And I wasn't deployed throughout the entire spring, throughout the entire summer, even at the beginning of the fall. When the winter hit, that's when this spike hit, and I got deployed. And I got deployed to deal with ill COVID-19 patients because they just needed doctors. They just needed people to deal with the patients. And I can tell you, I dealt with this disease as a researcher, as I described, but I'm not sure if I completely understood the gravity of the situation until I actually started dealing with the patients themselves and how dangerous this is and how prevalent it is and how impactful it is. I was particularly deployed to a part of the state that is underserved, does not have large hospitals, and has a huge population of patients getting this disease. And as I alluded to, and there is disease disparity, this particularly hits those that are socioeconomically challenged and disadvantaged. At the particular place that I worked, and I should tell you where I was deployed, remember I said that the hospitals are completely overrun. There are no beds. So I worked at a gym that was converted into a hospital with everything to give oxygen and drugs. And literally, we were building the plane while we were flying it, trying to create 21st century medicine in a gymnasium. And it was tough, but we didn't lose any patients simply because we were so attentive to their care. Now, I'm not going to spend time now. I can answer any questions that people might have about this, but 98% of the population that we dealt with was Hispanic populations from very tough backgrounds. There were lots of comorbidities. All the patients, many of the patients had diabetes. They had hypertension. They had kidney disease. They were obese. And I'll end with one little vignette that kind of tells a lot, I think, about what is going on here and what we need to do. So I took care of a patient who was symptomatic, and he told me, and to get into this unit, you definitely needed to test positive, because if you tested negative and you were in the unit, of course you would you would catch it. And if we're all wearing, these hazmat suits. And I was talking to this gentleman, and he told me his story when I was taking the history. He said that he had actually tested positive about 2 weeks before, but he had no symptoms. So he just went back home where his family and his extended family were living. Now, the first thing that pops into your mind is, well, if you tested positive, why would you go back home and risk exposing your entire family to this? And sure enough, they all caught COVID-19. His pregnant wife, his kids, the grandparents. But then the response was, you say, well, why didn't you quarantine? Or at least you think that. And the answer is, where would he go? Where could he go to quarantine? And I think that's part of the fate that we have. We have populations that, because of their circumstances, cannot help but spread it among themselves and among populations like this. So I think in addition to better drugs and better vaccines and better distribution, I think part of what we need is good old-fashioned help with public health measures, like providing places for people to quarantine, providing enough PPE for all the patients. So with that, I'll end, in both Sumit and I would be happy to take any kinds of questions. And I'll stop sharing my screen.
Thank you so much for those really fascinating presentations. And as you can imagine, we do have a number of questions. And so I'm going to try to combine them where they seem to work together and just work our way through as many as we can in the time that we have. So one question is, please speak to the South African variant and other possible other genetic variants, and the efficacy of the vaccine. And why do we think the COVID vaccines being distributed now will work for the new genetic variants? And after all, with the flu, we need a new vaccine every year.
That's a question that I assume it written all over it.
So, so the vaccines elicit what's called a polyclonal response. That means that they make a number of different antibodies. Your body makes a number of different antibodies to the spike protein in the virus. And so even if you have a single mutation or a couple of mutations, it's very unlikely that just a few mutations will evade the vaccine response. Now, that being said, it looks like the South African strain is going to be resistant to some of the antibodies that are being used as therapeutics, or they're going to be less efficacious. But you have, those are just single or double antibodies, whereas when we mount an immune response to the virus, there's tens and tens of different antibodies. And so even if you get rid of one or two of the antibodies, the rest of them will still take out the virus. So at this point, I think the virus will continue, or the vaccine will continue to be efficacious. Now, I think the $1 million question is really going to be, when enough people get vaccinated, there's what's called a selective pressure that's applied to the virus. Now, we haven't had enough vaccination to pressure the virus to form an escape mutant. These are just kind of happening by chance. But when you actually start pressuring the virus to mutate to survive this, the vaccines, that's, I think, when we are going to likely be seeing, or possibly be seeing, viruses that are could be resistant to the vaccine. The good news is that, like the flu, where we can change it up every year pretty quickly, we now have the technology with these mRNA vaccines to do the same thing. So if there's a new variant that's going around, we can quickly gear up, swap out the old variant for the new variant, and get people revaccinated. There's not really a limit to the number of vaccines you can take in your lifetime.
All right, thank you very much. Related question, the questioner is concerned about the country's new strategy to distribute one vaccine dose now and give the second dose later when available, even if it's more than 3 weeks later. And although it may provide some protection, how do we know what the immune system will do if the second dose is 3 months out?
Go ahead.
Yeah, so it's funny. I just got that question from someone in my family. Just WhatsApped it to me. The answer is we don't know. Theoretically, these vaccines right now, the mRNA vaccines are given with 2 doses, because the first one is thought to prime and the second one is thought to boost. Okay, so this is called a prime boost paradigm for vaccination. There are going to be vaccines that are coming online. The Johnson & Johnson vaccine is a single dose vaccine, so you don't always need the prime boost. The boost is thought to enhance memory functions of your immune system. So at least theoretically, it's not hard and fast that you have to give that within 3 to 4 weeks. You can slip by a few weeks, even a month or 2, and still see that boosting efficacy. Okay, but theoretically and actually having clinical data in hand are 2 different things. But I think there's a trade-off here. There's so many people dying that the opportunity cost here is that we know that the 1 vaccine dose can be at least short-term protective. Okay, and so the strategy here is to get doses into as many arms as possible, save as many lives as possible, and then come back and do the boost and see if we have long-term memory. If we don't, we come back with another vaccine, right? A single dose vaccine. If you took an mRNA vaccine, maybe you move to the ad vaccine. So yes, it's a risk, but I think, people are just weighing the cost benefit of giving it to people right now. It will save lives versus the potential dwindling of efficacy. We don't know what the answer is. I think the science tells us it's not going to be a problem, but the data is going to be what we need, and we won't have that data for another 3 or 4 months.
All right. The next question is asking about the safety of the vaccine. The questioner says, I've heard rumors of side effects such as infertility, and also that nanoparticles will be in the vaccine, which will illuminate under certain lights to prove that the vaccine was injected and that a person had the vaccine, and that it will be mandatory for future air travel.
I can answer. I have been vaccinated. I had to get vaccinated to take care of the patients, and I've not experienced any of those problems. And even I've stood in front of a black light and I don't glow.
Okay. Comment on that?
Yeah. So they don't cause infertility. There's some safety signals. Most of them are based on the reactogenicity of the vaccine. So people who have a history of severe anaphylaxis should either talk to their doctor or at least have some Benadryl or an EpiPen when they go in for the vaccine. Beyond that, the vaccine is not a permanent thing and actually dissipates within weeks, right? This is an mRNA vaccine. mRNA does not hang around. Most likely what it does is it goes to your liver, makes the spike protein, your immune system recognizes it, mounts an immune response, and it gets flushed out just like a virus would.
Okay. Here's a pair of questions that I think you've largely addressed, but just in case you want to say a few more words about them. One question, how long does the vaccine last? Will we need another shot in 2022? And then with respect to the allergic reactions, what is considered a severe allergy enough not to have a Pfizer vaccine? Can allergic levels be tested in advance? Is epinephrine enough to save you? And how long before an allergic reaction would manifest?
Evan, that's a MD question.
I think that, I think the concerns for this vaccine are no different than the questions one asks about any vaccine. And most of us have been vaccinated for all kinds of reasons. If at a particular age, we got MMRs and we all get flu vaccines every year. So the risk of an allergic reaction for this vaccine is no greater than for any vaccine. If there were to be a severe allergic reaction, it would be fairly immediate. And Benadryl and epinephrine certainly are adequate. Most of the time, even if there's a little bit of irritation, and as I said, I was vaccinated along with my whole cohort of providers, and which is, there were about 120 people at our installment. And, the most that anybody experienced was a little bit of redness around the site, maybe a little bit of itching, and nothing severe.
Okay, going back to the issue of virus infection issues, what makes the new genetic variants spread more easily? Is the virus's spike protein longer, stronger, et cetera? Or in what other ways does the virus infect the cells if not via the spike proteins? A couple of questions there.
The short answer is we don't know yet. So most of the transmission data has been based on epidemiological tracking and sequencing of the variants. So this variant seems to be outcompeting the other variants. And so the most plausible explanation is that it's better at either infecting cells or being transmitted from person to person. But the experimental evidence hasn't been really rendered. But it's likely going to be mutations in the spike protein, if I had to bet, that allows this virus to either enter the cells, replicate at higher levels, or just be more stable in transmission. But the jury's still out as to what the underlying mechanism is.
And I can reiterate that this is an active area of research. In some of the long organoids that we've looked at, there are ways for the virus to enter cells independent of the well-known receptor. Some of you may have heard it called the ACE2 receptor. It can get into cells that have different receptors or not that receptor, but that also underlines the importance of not only continuing research, but combining not only immunization with certain drugs that we know will block alternative routes of entry. So I think ultimately these diseases, particularly if they wind up escaping vaccination, we'll need a kind of a cocktail of drugs to block various kinds of symptoms or block spread from cell to cell that might employ other mechanisms. But I think we're watching that. We're not going to be taken by surprise. I think we're carefully looking for all of these.
Here's a more long-term question. Do you suspect that we will see a lot of health problems down the road, maybe 10, 15, 20 years down the road in people who were sick but recovered? And maybe you can just sort of generally address some of those issues that have been reported already.
Yeah, I mean, there's these long haulers, right, that continue to have long-term effects of COVID. So we don't know what we don't know at this point, and what COVID does, what SARS-CoV-2 does to the body beyond just the lung inflammation is an active area of research. Evan talked about it, and maybe Evan, you can jump in here. But I think that there are going to be complications from COVID. Even if you did recover, you're going to have different complications for different people. And these are going to be sticking around for a long time. And we're going to have to make sure we try to address those complications. Evan?
Yeah, Sumit's completely right. We don't think that this virus is like a herpes virus, for example, or a varicella virus that once you get infected lives dormantly throughout life, causing problems. But it could, for example, and again, we're still trying to understand what it is that the long haulers are complaining of, what is their disease. One thing that we are looking at and we are aware of is problems caused not so much by persistence of the virus per se, but the kind of damage that the virus did when it was active. So remember I said that we think, and we didn't think this initially, but now we're starting to think more and more, that this is a heavily a blood vessel disease. There's a true vasculitis that can cause leakiness in the blood vessels, but also clots in the blood vessels. Clots in the blood vessels are what cause heart attacks and strokes. So there could be some persistent problems if during the infection there were many strokes. And then you're dealing not so much with a persistent viral infection, but the sequelae of having had a stroke or something happened to your heart that's secondary to the viral infection, but one is dealing with that. This is still being investigated very assiduously, but I can tell you that because of our concern about this being a vascular disease as well and an inflammatory disease, every single patient that we took care of got placed on dexamethasone, which is heavily an anti-inflammatory drug, in addition to any of the developmental things I talked about. And they all are placed on anticoagulants and antiplatelet drugs to try to prevent these kind of sequelae. Every patient, that's just routine.
Thank you. So I think it's time to wrap. Let me read this final question.
I have a question. I didn't see it on the questions over here. Could I ask it?
Well, can you put your question in the chat box?
I did, and it didn't. It's down here on the bottom. Vitamin B3. What does it do? And it didn't go up with the questions.
Oh, okay. Why don't you tell us quickly what your question is, please?
The vitamin D, the supercomputer UCSD came off with vitamin D3 has an effect on whether you contact or get ill with this virus. What does it do? Vitamin D3.
So I've seen reports of vitamin D3 having some clinical benefit, but that hasn't been. It's been largely anecdotal. So vitamin D3 will obviously help your immune system.
I'm going to give you an experience. My
Yes.
sister-in-law, 87 years old, where the home where she is, they've been giving her vitamin B3 now for 4 months. Okay, they found in her blood the viral effects of COV. She had it, but her symptoms, she had a little diarrhea and that was it. And she's 87 years old.
All right.
So it had an effect.
All right, thanks. So let's let Dr. Chanda wrap on that question, then I'll have one closing question and then maybe a final comment from each of our speakers.
So I mean that's exciting, but generally what we need to do is do what are called case control studies, where you give a 1,000 people a certain drug, and so it's very hard.
8 years old, so I was interested.
Yeah, no, I mean, listen, it doesn't have any harm, right? I mean,
Right.
if you take I've
been on vitamin D3 now for 3 years.
But it's unless it's been really proven, using
Right.
these kind of case
I know what
control
you're
approaches.
saying. Yeah, I know what you're saying, but
Yeah.
still.
No, there's a lot of things that are out there, and it's our job to kind of sift through them and see which ones work across the population, across different people, and there's science behind it, and this is one of those.
And I read in that research also, that page. Could I just say this?
Jean, let me just get to the final question, if you don't mind, because we're at the end of our time. So the final question is this: can you talk about herd immunity and how this relates to the need to continue to vaccine like we do today with the seasonal flu? If you could address that question and then offer whatever closing remarks you have. We thank you for your generosity with your time and your expertise, and this has been a very interesting program.
Why don't you answer the herd immunity, Sumit?
Sure. Herd immunity goes back to this R naught equation that I talked about. So herd immunity is essentially where either enough people have the vaccine or have been infected where the virus doesn't have anywhere else to go. And so that R naught goes below 1. So for every person that gets infected, it gets transmitted to fewer than 1 person. And so that's where you see the curves of the virus, instead of going up like it's going now, it's going down. And so that herd immunity we're predicting is going to be somewhere between 70 to 90% of the population. And so herd immunity gets us out of this pandemic. Okay. Flu is seasonal. And it also changes every year. So we do not have herd immunity against flu, which is why we need to keep revaccinating every year. But we have enough immunity to flu where it doesn't rage rampant throughout the year until we get a new variant of flu, like the swine flu, and that's what causes the pandemic, right? There's no pre-existing immunity, and it's just fire ripping through dry timber, right? Whereas herd immunity is a point where there's not enough places for the virus to go where it doesn't cause the pandemic, but don't confuse that with eradication. Herd immunity is not eradication. Herd immunity means life goes back to normal, but we live with SARS-CoV-2, and we have to keep vigilant and keep vaccinating and keep coming up with drugs.
Thank you. Final comments from our speakers?
I guess I would just end by saying that this is a very fast-moving field. And as Sumit alluded to, there are known unknowns and then unknown unknowns. And we're constantly being surprised at the things that we need to learn. But I think that investing in research, investing in better public health, and then trusting the science, letting science dictate what we do is probably the path forward. And I think we will get through this as we've gotten through so many other kinds of public health challenges. If we just invest in data and the research and protecting our community.
Yeah, I can't echo that enough, right? Science is apolitical. It doesn't have a party, and it's driven by data. And so it's very important that the people trust the scientists and public health experts and listen to their advice. We are in the most dangerous phase right now of the pandemic. Even though there's light at the end of the tunnel, right now we're going to continue to see 4,000 deaths a day across the U.S., and that is just a staggering number. Unless people continue to be vigilant, more vigilant than they are being right now, continue to mask, continue to stay social distance and stay home, it's going to get more horrific before it gets better. So as Evan said, we have to listen to the science, we have to listen to the public health experts and minimize the suffering, the pain, and the loss. So then when these vaccines do become available to everybody, we will not have paid the huge price that we're paying right now.
All right. Thank you so much to Dr. Chanda, Dr. Snyder for this very informative program. I thank all of you for your attendance and your questions and your interest in this. And this video will be on our website, the delmarfoundation.org, within the next day or two. And so you can refer your friends and neighbors to see it there. Thank you all again for attending, and wish you all a good night.
Thank you. Stay safe, everybody.
Stay safe. Thanks for inviting us.